The next speaker, Daniel Priego Espinosa. He's a PhD student of the program in Biomedical Sciences of the university, and he works at the Institute for Physical Sciences in Cuernavaca, and he's going to talk about scaling networks related to fertilization.
Well, thank you for the introduction. First of all, I'd like to acknowledge my collaborators and supervisors. Adan and Dr. Adan and Dr. Alberto Darson, they work in Institute of Biotechnology in campus Cuernavaca. Gustavo Martinez Meckler, he is my supervisor. And we have a strong collaboration with Jorge Carneiro from Instituto Gulbenkian de Ciencia in Portugal. Why we're studying is, we are studying external marine fertilization, and especially in sea urchins. Sea urchins are an excellent model for more than 150 years, and fertilization is not the exception. This is one of the species, Strongylocentrotus purpuratus. And just as many marine species, they reproduce by a broadcast spawning, which is no other than an orgy in the sea. So this is like a marine porn. Males and females, they release their gametes into the sea, and once they are released, they have the challenge to encounter each other. Just to know, the males release up to 10 to 11 sperms per emission, and females release up to 1 million eggs per emission. So this amount of cells might be considered as too much, but females and males, they don't release these gametes precisely at the same time. There may be some predators going on. So this is why, given that this is external fertilization, it's very easy to reproduce the environment in a laboratory. So, to overcome this dilution effect, eggs attract the sperm by means of a chemoattractant. This chemoattractant can be synthesized in the lab, and you can precisely on photo cleavage and release active this molecule. So this is a movie of a bunch of sperm, and you can, just by releasing by ultraviolet light, you can point a center of release. This is RESAC, the name of the chemoattractant, and you will see an aggregation of sperm in the middle. But how this is changing the behavior of a single sperm? So as I told you, these molecules which come from the egg, from the outermost layer, these are diffusible small peptides. They form a gradient on the egg, whereas sperm, which are in resting state, they are swimming around circles in 2 dimensions or in 3 dimensions as a helix. Once they start becoming stimulated by these peptides, they change their behavior to a series of interspersed acute turns followed by straighter trajectories. If the timing is correct, this sperm will in the egg. This is the sequences of 2 of the most used sea urchin species. But how is this being controlled? This is controlled by a calcium signal triggered inside the flagellum of the sperm. This is the same experiment where you can see just a movie of a sperm stimulated by a sap stimulus. Sorry, I have to reproduce it again. This is going to be, this is a sperm swimming around, then it becomes stimulated, and you will see a signal of red. This is by a means of a fluorescent reporter that binds calcium. So as you can see, every time there is an acute turn, the signal of calcium will increase. It has been reported that the increasing calcium rate inside the flagellum is correlated to a higher path curvature, whereas a decreasing calcium rate is correlated to the low path curvature. So, the temporal features of this calcium signal is regulating the sperm swimming, but this implies a synchronized, a recurrent feedback between two kinds of variables. The external variables are given by the periodic stimulus that is encountering the sperm as is approaching the egg. So there is a shape of shape of the sap gradient and how it's swimming in this gradient. The internal variables are all the set of molecular events in the signaling network that will ultimately lead to calcium oscillations. I forgot to tell you, SAP. SAP is the generic name of all the kind of peptides that are attracting the sperm, because this is species specific. So, even though I was talking that this implies a temporal signal, this translation of a spatial pattern into a temporal calcium pattern by the sperm. In the laboratory, we mostly used a simpler experimental setup, which is we released a determined amount of these peptides. There is no changing. There is no changing. Yes, there is no change in time of the stimulus, and even though it's a fixed stimulus, you can see some spiking trains, some calcium spike trains. These are just some examples of how are these at the single cell level, how these calcium signals in the flagellum develop through time. So we quantified two essential properties of this signal, which is how is the interval between the peaks. The other one is how the amplitude of these peaks is changing in time. So as you can see, this is just a label of numbering of each of the peaks, of the calcium peaks, and you can see that the interval of this interval is increasing with time, whereas the envelope or the amplitude is decreasing in time. And this is just a statistics of hundreds of recordings. How is this response being regulated by these peptides? Well, I have to mention that sperm are translationally and transcriptionally silent. This implies that there's no turnover of all the protein components. So they are born with a definite setup of proteins, and these will not change throughout time. So this is a as lack of the flagellum, and this is mainly controlled by a set of ion channels, which are controlling the voltage. The SAP peptide is binding to a receptor, which itself has a one-late cyclase activity. There's also some pH changes. There's an intertwined set of molecular events, which in the end will control these calcium spikes. Nevertheless, we are interested in knowing which are the calcium channels that are allowing the influx of calcium in the sperm flagellum. There has been amount of evidence for many types of calcium channels. This is the most recent one, Catsper. These two ones are related to the classical voltage-dependent calcium channels, the same type of channel that you would encounter in nervous cell system and other cells. This Caspar channel, which is more recently discovered, is a sperm-specific channel. However, the evidence or the type of evidence for reporting this relies only in germ cell precursors, not in the sperm per se. There's also some pharmacology dependent evidence. And I have to mention that this organism, contrary to mouse, which you can genetic engineer to do some knockouts, this is not the case. So we only can rely on these kinds of drugs and toxins. So there's this caveat that the available blockers are never 100 specific. Even though they report that this is specific for this kind of channel, they can cross-react to other channels. So this is a challenge to know exactly how these channels are controlling the response. So we proposed a kinetic model for this signaling network, which is composed of an upstream module, which has to do with the earlier response. There are two modules which are exploring as alternative scenarios. Which one? This one is centered on this classical voltage-dependent calcium channels. This is only dependent on voltage, whereas this Caspar module, it has a threefold dependence. It depends on voltage, on calcium itself, and the pH. So we are working with an ODE system, a system of differential equations, and these are the observables that we are to compare to measurable physiological variables, which are the membrane potential, the cyclic GMP, calcium, and the protons. So this is the kind of equations that we are using for modeling the receptor activation for SAP and the cyclic GMP dynamics. This is a contribution by the active receptors. This is the membrane potential dynamics, for which we are following an Hodgkin and Huxley-like formalism, which it depends on the sum of the ion currents. The ion currents are by these Ohm equations. We have a leakage current term for fixing the resting membrane potential. This is the calcium dynamics, and this is the proton dynamics. So we first calibrated the upstream module, for which there is plenty of information at the level of a cell population. So this is a calibration for the kinetics of cyclic GMP. This is an experimental setup of several SAP concentrations, and these come from millions of cells, and we are fitting the model for this. We have also membrane potential dynamics in cell population, and we adjusted these the early membrane potential response, which is a hyperpolarization. As you can see, a drop in the membrane potential, and then there is a slight over, even though we are here dealing only with two channels which have opposite actions. This one will hyperpolarize because it allows the outflux of potassium, whereas this allows the influx of sodium. So this is enough to explain the very early steps of the cell signaling network. Afterwards, we started calibrating independently the modules. This is the CAF module, This is the kind of dynamics that we can generate for the calcium cyclic GMP. It's not showing us as sharp, but we have the channel gating, the current densities, and the membrane potential. This is for the Caspar module. which I have to remind you that it has a threefold dependence: voltage, calcium, and pH. This is a kind of response that we can find by parameterizing the model. So as I told you before, we are interested in trying to reproduce two of these essential features of the calcium response, which are the envelope, the amplitude of the calcium spikes, and the interspike intervals. Here I am contrasting the two dynamics by the CAV module and the Casper module. If we measure the interspike intervals, you can see that they are, this is an overlay with experimental analysis, they are following more or less the same trend. So up to here, they can both explain these essential features. Since I'm working in a dynamic, in a lab with physicists, we try to do dynamic system approach, which is doing a bifurcation diagram of the two modules. This is to see which are the capabilities, the possible capabilities of these signaling modules. Here, the parameter of bifurcation, we are transforming the cyclic GMP, which is one of the first signals. So we are changing this variable into a parameter. So each of these in this axis would imply that time is fixed with a certain concentration, and we are measuring two properties. In this first row, we are measuring the steady state, the asymptotic state of calcium, and in this lower row, we are exploring the periods of the periodic regime, if there is any. So for the CAF plus BK module, you can see that there is some bistability. These two bistable states are periodic. There are two limit cycles coexisting for this module, whereas for the Casper module, there's only one, and actually there is a sharp transition in these cyclic GMP ranges. There's also a bistable regimen around the resting state of cyclic GMP. We still don't know what this means, because calcium, which is a very low at the resting state, the system would be here, but in principle, it's also possible to reach this high calcium state. There's also, you can see, an increasing interval, an increasing period for both models as the cyclic GMP diminishes. So one alternative to evaluate how well these two models would explain, one is altering by external stimuli like alkalinization by ammonia. This noisy signal comes from the experiment, and we are overlaying the simulation results. This is equivalent as clamping the pH at a certain amount as of 5 seconds, and you can see that the Casper module is explaining well the results. Okay, this would correspond to the mixed scenarios, but apparently if we combine the two modules, they don't behave too well. So these are the conclusions, that both CAP and Casper can qualitatively reproduce these two main kinetic features, the envelope and interspike spacing. And there's multi-stability in different ranges, indefinite for these two modules. One is two periodic regimens, and the other one is low and high steady state around resting state. This experimental test of fixing pH to high levels with ammonia indicate hints to distinguish these two scenarios. There are more predictions by the models, but these are still to be tested in the laboratory. Well, here we are still working with these with the coexistence of these two modules, but this is still ongoing work. And thank you.
Questions?
Thank you. It's probably a very basic question, but thinking about the two navigational strategies, the sperm take, one is going straight and the other one is the spiral one. What is the motivation to switch to the spiral one that seems to have a longer trajectory? Is it also moving faster while it's doing that?
No, actually the sperm is not swimming. The stereotypical pattern is composed of these periods of acute turns, but they are followed always by a straighter swimming. They are not changing to always straight. This is seen more in mammalian sperm, but for marine fertilization is not the case.
So it's kind of like a run and tumble where the...
Yeah, kind of,
okay.
yes.
Another question.
Thank you. Between those two models that you showed for the results section, the first one captures the profile better, right? The envelope is much better in the first one, but the second one has the better performance in terms of the
interspike
right?
interval.
Are you working to combine them together so that the envelope of the second model also can get better taper?
Yes, well, already disconnected, but by my explorations is that adding a small amount of the CAF channel to the CATSPER module, you increase the frequency without altering the envelope. So the message is that CATSPER is the main channel, the main calcium channel, but you can still tweak the frequency by these secondary channels.
How about the amplitude?
The amplitude?
Yeah, the amplitude also goes down in experimental data, right?
Yes.
Yeah. Did you create that kind of diminishing envelope?
Yes, it's creating this kind of a diminishing envelope.
Thank you.
Yes, this is also a very basic question. There are two kinds of sperm, right? X and Y sperms.
Right.
And what I've heard, and I don't know if it's true, is that one of them, probably the Y, is faster than the X, and that the X is more resistant to time.
Yes.
Is there any difference in energy transfer or whatever in the calcium channels between X and Y sperm? Because if one is faster or whatever, probably it's spending more energy, right?
Well, that's an interesting question, but this is a low Reynolds regimen. So here the acceleration is not so evident. Well, a short answer would be those would be negligible effects for the amount of energy. It would be interesting to do some calcium, some experiments and some estimations, but I wouldn't find it as an important factor for the amount of energy in this. In the experiment, in principle, yes. By this gradient precipitation, you can do some. But along these trajectories, no, there is no way to distinguish them.
Question? Well, I have one. Have you actually seen these bifurcations? Well, your model predicts bifurcations and bistability and things like that. Have you actually seen those in the experiment? Is there any evidence that there is a bifurcation?
Well, that you would see it, for example, here in the bifurcation diagrams. I chose the cyclic GMP as a parameter of bifurcation because in the experiment you can, on cage cyclic GMP, you can stimulate and bypass the sap binding to the receptor. So an encouraging result is that if you increase too much of cyclic GMP, you could still see an increase in calcium, but not a spiking trend. If you go lower cyclic GMP levels, you would find this periodic regimen. If you go very low near the resting state, you wouldn't see any response. So that would be a hint of a bifurcation.
Okay. If there are no more questions, let's thank the speaker again, please. Now,